Cell Cycle Part 3, and we will discuss about stem cells in this part. You can't really talk about the cell cycle unless you talk about stem cells and talk about cloning. And so in this part, we are going to focus on stem cells, and in the fourth part, we are going to talk about cloning. So why is everybody so interested in stem cells? Why are they focus of so much attention in the scientific research community? Why are they often in the news? So in developing countries, in the third world countries, most individuals die of infectious disease or malnutrition. But in developed nations like our own, the most important diseases are degenerative diseases because of improved hygiene and improved medical care. Infectious disease malnutritions are not as much of a problem as degenerative disease. And here is a list of top 10 leading causes of death in the US. And this comes from a CDC report that was published in 2017. And you can see that I have labeled with asterisks the top 7 of the top 10, which are degenerative diseases. The number 1 killer in the US is still heart disease, but not far behind is cancer. And in fact, it's predicted that within the next few years, the cancer may actually surpass the heart disease and be the number 1 leading cause of death in the US. You have chronic respiratory disease, cerebrovascular disease such as stroke, Alzheimer's, diabetes, and nephritis. So these are all degenerative diseases that are the curse of the developed nations. However, some good scientific data suggest that stem cells may be the answer to some of these degenerative diseases. So in support of how rapidly stem cell research is now, or how important it is now, the Nobel Prize for Physiology or Medicine in 2012 went to 2 individuals who did quite a bit of work to advance the field of the stem cell research and advance our understanding of stem cell biology. The first, and in this shared Nobel Prize, the first one was John Gurdon, who pioneered a technique known as somatic cell nuclear transfer, which we are going to talk about in the fourth part of this subject. But essentially, he cloned frogs using this technique. And this told us a lot about the stem cell biology, which I'm going to elaborate on in a minute. Dr. Yamanaka demonstrated that there were 4 genes whose expression was critical to giving an embryonic cell its phenotype or its embryonic stem cell nature. So he took adult cells from both mouse and human, and he turned on the expression of these 4 genes listed here, which are the OCT3 and 4, SOX2, KLF4, and c-MYC. And he successfully turned adult cells into embryonic stem cells. Today, these cells are produced by this technology are known as induced pluripotent stem cells, or iPSCs. Again, this was a very prominent and important finding, and these 2 individuals shared the Nobel Prize. So there are 2 properties of the stem cell that make them very interesting. The first is that they can propagate themselves. Okay, so when a stem cell divides, it sometimes referred to as an asymmetric cell division, because one of the cell is another stem cell. So it propagates itself. It keeps itself going. The other cell is the daughter cell, might be a progenitor cell on its way to become a differentiated cell. So the first property, so that it can propagate themselves and maintain themselves. The second property is that they can be differentiated. For a long time, it was thought that once a daughter cell embarked on its path towards a differentiation, it could never go backwards. It could never become the stem cell again. But both of those individuals who shared the Nobel Prize showed otherwise. Dr. Yamanaka showed it by expressing those 4 genes, but Dr. Gurdon showed it because what he did was he took a nucleus from an adult frog cell and put it in an enucleated egg cell, and it developed into a tadpole. So that showed that you could actually reverse this process. And that's the importance of their findings, and that provided significant insight into the biology of stem cells and the biology of differentiation in general. So some stem cells are more plastic than others. What I mean by that is that some can be differentiated into a lot of different cell types, But then some cells that are maybe farther along in this pathway can only be differentiated into some cell types. So the plasticity of the stem cells usually is classified as potency. A stem cell that is unipotent can only form one type of differentiated cell. A stem cell that is multipotent can form multiple differentiated cells, and there are about 200 different cell types in the human body. So a stem cell that's referred to as pluripotent can form all of the 200 differentiated cell types that you normally find in the human body. And then a stem cell that is totipotent can form all of these differentiated cells plus some of the specialized tissues like those of the placenta. Okay, so let's look at some of the sources of the stem cells. So let's take a look at the adult stem cells and embryonic stem cells as well. So for decades, it has been known that there are stem cells in the bone marrow that can form all of the cell types that you can find in the blood. For an example, there is a pluripotent hematopoietic stem cells that is called pluripotent, but it's more like a multipotent cell, which is probably a more appropriate term. It can be differentiated into a lymphoid lineage, and the lymphoid lineage will give rise to the T cells and the B cells, which are really important cells of the immune system. Or they can be differentiated into myeloid lineage and give rise to platelets, red blood cells, eosinophil, macrophages, granulocyte, and the last three cell types that I mentioned here are also really important in the immune system. So bone marrow as a source of adult stem cells, and for decades that has been known, so this is nothing new. The structure of your intestine is a very convoluted epithelium that is made up of these villi and the crypts. And also for several decades, it has been known that there are stem cells or intestinal stem cells that can be found in the crypt region of the intestine. And you know that epithelial layer of the intestine is turned over quite rapidly in about 3 to 5 days. All of these stem cells that become very rapidly differentiated and move up the crypt into the villus to replace some of these epithelial cells that are on the surface of the villus. So right in this region here in the crypt is the stem cell region. There are these stem cells in the green, and these are called the LGR5 plus stem cells. LGR5 plus is a leucine-rich G protein coupled receptor 5, which is a biomarker of these intestinal stem cells. Means these receptors are only expressed in these stem cells. The dark blue plus 4 stem cells are the reserve stem cells that occupy the 4th position from the crypt base and generated from LGR5 plus stem cells, and can restore the LGR5 plus stem cells compartment following an injury. Then in the medium shade of the blue, you have the transient amplifying cells that divide to produce a variety of differentiated cells like enterocytes, tuft cells, goblet cells, enteroendocrine cells, and these are all shown in different colors. And as they proliferate, they move up and they migrate and replace the epithelial cells in the villus. But this yellow pannate cell migrate downward at the base of the crypt and secrete antimicrobial defense proteins. So you can actually track this microscopically. So here are 2 intestinal villi, and this is an intestinal crypt. And here are some of these stem cells that are labeled with this dark black dye. And at 40 minutes, they are starting to move up the region. 2 days after labeling, they are almost at the tip of the villus. And then about 3 days after labeling, they are at the top of the villus. And again, this is nothing new. These stem cells have been known for quite some time. Another source of the adult stem cells, and the nomenclature is a bit confusing, but they are called embryonic carcinoma cells. But embryonic carcinoma cells are not embryos. These cells are derived from tumors that arise in germline cells, so from sperm and egg. So sometimes sperm cells and egg cells can lose their cell cycle control and they form these tumors. And when these tumors arise from these germline cells, the cells in them are called embryonic carcinoma cells. And when one of these tumors is benign, it is known as a teratoma. And when one of these tumors are malignant, they are called teratocarcinomas. So here is a picture of the testicular teratocarcinoma, and it's a malignant tumor that has arisen from a sperm cells. These are very interesting tumors because within the tumor, you get differentiation into different cell types. So you can see that in this testicular carcinoma that you actually have hairs growing. So these are very unusual tumors. And in this X-ray is an individual that has an ovarian teratocarcinoma. So this is a tumor that has arisen from an egg cell in the ovary, and it actually has teeth. So there's a differentiation of teeth, and you probably are familiar with the Lance Armstrong, who was diagnosed with a testicular cancer, a testicular teratocarcinoma. So there's been a lot of work done on these embryonic carcinoma cells. They are very plastic. And here is an example of one of these experiments. The embryonic carcinoma cells, given the right cues, can be differentiated into a lot of different tissues, including neuronal cells. So here's some embryonic carcinoma cells that you treat with them with 10 millimolar of retinoic acid for 28 days, and you get the formation of these neuronal cells. So you can see that they are very plastic and they are very useful model for studying stem cells. So another source of stem cells is the embryos, and these are the embryonic stem cells, and of course they are the ones that are controversial. So when you have a development of the fertilized egg in 2.5 days, you have this 8-cell stage. In about 3 days, you have the 16-cell, and eventually you have the formation of the blastocyst. And inside this blastocyst is a group of cells known as the inner cell mass. And these are the embryonic stem cells. So if you want to grow embryonic stem cells in the lab, you would dissociate the cells of the blastocyst and you would collect these inner cell mass cells, and then you would put them in a culture dish. And embryonic stem cells have very fastidious growth requirements though. You remember that at the beginning of the course, we talked about given appropriate growth requirements that you can grow just about any kind of cells in culture as long as you give them appropriate vitamins and serum. Embryonic stem cells actually have another layer of requirement for growth. They have to be grown in the presence of other cells. And so usually when the scientists are growing embryonic stem cells, they will have another layer of feeder cells. They are usually the fibroblasts, and they are irradiated. And the reason that they are irradiated is because that halts the cell cycle, that damages the DNA. And remember, we talked about the DNA damage checkpoints. So you have a damaged DNA in those fibroblasts so that their cell cycle is halted. They're alive, they're growing, they are secreting the factors that are necessary to grow the embryonic stem cells, but they are not dividing. So they won't take over the culture. So you have these embryonic stem cells that are growing on this layer of irradiated feeder cells. There are two controversies that have been associated with these embryonic stem cells. And the first is that to isolate them from the blastocyst, you necessarily destroy the blastocyst. And this is a huge question in the bioethics, whether this is morally acceptable to destroy blastocyst in order to isolate embryonic stem cells. The other controversy is the growth or the co-growth with these fibroblasts. And some people do a lot of measurements on these embryonic stem cells, and they look at the physiology of them in this condition. And some people question whether you are really looking at the embryonic stem cells or whether you are looking at the fibroblast. And when you passage embryonic stem cells, are you really passaging some fibroblast? Or are all fibroblasts truly irradiated in the way that they cannot grow? And so there is a contamination of embryonic stem cells, possible contamination with fibroblast, which can confound your interpretation of any kind of data that you get from them. However, the problem of the feeder cells has been solved somewhat with the use of cytokines instead of feeder cells. But again, this has been a problem in the embryonic stem cell research. So two controversies, one of technical or scientific controversy, and the other is bioethical controversy associated with embryonic stem cells. So it has been reported or purported that the embryonic stem cells are pluripotent, which means that they can form 200 cell types that are normally found in the human body. And there are three bits of evidence that support the hypothesis that embryonic stem cells are pluripotent. The first is that under normal developmental conditions, embryonic stem cells form all of the tissue of your body. Of course, if the blastocysts are allowed to fully develop, you end up with all the tissues of the body. So that's a pretty good evidence that embryonic stem cells are pluripotent. The other evidence is that if you take embryonic stem cells and you inject them into immunodeficient mice, you form teratomas. And if you look very carefully at those teratomas, you will find all kind of differentiated cells from different tissue types, from the ectoderm, from the mesoderm, and from the endoderm. As you can see that the tissues that fall under these categories. And so in those teratomas that are formed by the embryonic stem cells that have been injected into immunodeficient mice, you find all those tissue types which suggest that the embryonic stem cells were originally pluripotent. Then there is another interesting experiment, visually interesting experiment. You can do that to support the hypothesis of the embryonic stem cells that are pluripotent. So here is a mouse blastocyst, and you isolate the embryonic stem cells and you grow them, and then you transfect them in such a way that they now have a GFP tag. So now they're green. When you take those green embryonic stem cells and you inject them into the blastocyst of another mouse, and then you take the blastocyst and you implant them into a surrogate mother, when you will notice that the offspring will show the GFP fluorescence in practically every tissue in the body. So here is the blastocyst. You can see the GFP cells within it. Here is the whole embryo that is green. You can see the green, like I said, in every tissue of the body, and that supports the potential pluripotent nature of the embryonic stem cells. What is the hope for embryonic stem cells? So in vitro, there has been some success at differentiating embryonic stem cells to all the different tissues of the ectoderm, mesoderm, and endoderm. Note in vitro or in cell culture though. So many people are conducting experiments to find the right condition to differentiate an embryonic stem cell into practically all of these tissues in the body. And there has been some success, as I mentioned, getting embryonic stem cells to become these kind of cells in culture. This is shown here on this chart. I want to point out again that in culture or in vitro, there has been some success. But in vivo, there has been very limited success. So if you take the embryonic stem cells and inject them into a mouse, you will probably going to get a teratoma. But there was one research group that did an experiment where they differentiated embryonic stem cells in the culture dish into hepatocytes, which are the liver cells or the cells that looked like a liver cell based on the physiology and on the genes and the proteins that were expressed. This group also added a GFP transgene so they could track it. And if they inject those differentiated cells into a mouse, and the GFP hepatocytes that had come from the embryonic stem cells populated the liver and persisted in the liver and seemed to behave like liver cells. But to date, the success of embryonic stem cells populating an organ and behaving like the cells in that particular organ have been virtually zero. So, the clear advantage of embryonic stem cells is that they are pluripotent. Here are some of the disadvantages. You almost always get a teratoma formation, and I didn't mention this, but it is difficult to culture them. It is difficult to establish cultures. It is very important that maybe only 1 in 10 embryos provide usable embryonic stem cells. When you differentiate them, even in the culture, you often get a mixture of cell types. So in that experiment where they differentiate into embryonic stem cells, hepatocytes, they didn't all differentiate into hepatocytes. Some other cell types were there as well. There is limited evidence right now that embryonic stem cells might be useful in treating degenerative diseases. There is a whole issue of the transplant rejection. So you still have that problem. So if you take embryonic stem cells from one animal, for example, and you differentiate them in culture and put them in another animal, it recognizes those differentiated cells as foreign, and you can get transplant rejection by your immune system. There is some evidence that the embryonic stem cells and differentiated cells have unstable genome. And of course, as I mentioned at the beginning, that there are some ethical questions. So these are some of the disadvantages of embryonic stem cells. But the adult stem cells, there has been more success in vitro. There are reports of treatment for more than 90 medical conditions and diseases, including sickle cell anemia, multiple myeloma cancer, and damaged heart tissue. As you can see here, the adult stem cells have been successfully differentiated into many other cell types. For example, stem cells from the fat tissue have been differentiated in culture into bones, into cartilage, into muscle, into neurons. Stem cells from the brain have been differentiated into brain cells, nerve cells, muscle cells. So you can see that there's been quite a bit of success in culturing and differentiating adult stem cells in culture or in vitro. And in vivo, there has been a little bit more success with the adult stem cells than the embryonic stem cells. Adult stem cells have been successfully used so far to treat spinal cord injuries and with some success to regenerate heart tissue, to treat muscular dystrophy, diabetes, Crohn's disease, Parkinson's, and hearing loss, to regenerate cornea and hair loss, and some cancers as well. So more success in vivo with the adult stem cells. There are some advantages and disadvantages of adult stem cells. One of the advantages is that they have less fastidious growth requirements. So for adult stem cells, you don't have to grow with that layer of irradiated feeder cells. They multiply indefinitely. So I didn't mention this, but embryonic stem cells seem to have only a certain lifetime in culture. And then after that, they stop dividing. With adult stem cells, you can do autologous transplants. So for example, you could take someone's bone marrow and differentiate in culture and then put it back. And so you overcome the transplant rejection disadvantage that you see with embryonic stem cells. One disadvantage that has been reported for a long time is that they are less potent. But given the recent advances, especially in the research of Dr. Yamanaka, who showed that you could take an adult stem cells, turn on those 4 genes, and it becomes embryonic and pluripotent, thus making this disadvantage not a big issue anymore. So this completes the part 3 of the cell cycle and our discussion on stem cells.